Hey guys, so here we are back in nutrition science. You guys are on the taster lab, and we are trying to determine somebody's ability to taste. So you watched a few videos, but we want to talk about the genetics end of what's going on over here. So we talked about a few different things. We said that somebody can be what we call a taster. There were a bunch of different terms, but we'll just say that someone can be a medium taster, and someone can be what we're going to call a non-taster. So when we talk about this from the genetics end of it, this is, if you remember your genetics terms, this is the phenotype. This is the way that the specific trait is expressed, how we see the trait, like brown eyes would be a phenotype. So we need to assign a genotype to see how this will be expressed in the population. So we need to use letters, which we call alleles. So I can tell you that being a taster is a dominant trait. So if we assign the letter T to this trait, then if it's a dominant trait and being a super taster is dominant, we're going to use a capital T. And if we know that being a non-taster then is a recessive trait, then we're going to use a lowercase t. So we know that to each genotype, right, the way the genes look, we need to associate two alleles to those. So you have a couple of different possibilities, but in using the medium taster, we're going to kind of introduce it this way. So to be a taster, remember, to show a recessive trait, you have to be what we call homozygous. Both alleles have to be the same. So therefore, little t is going to be how we express that particular trait. Now, the super taster then is going to be homozygous as well, and we're going to have that being expressed as a capital T and a capital T. So the medium taster, which is, again, in between, we're not really going to be able to know the difference. When we do this test, we're really going to know the difference between a taster and a non-taster. But the medium taster, then, we're going to make that a blending, and we call that a heterozygous trait. So that's going to be expressed in that way. So these are our phenotypes, and these are our genotypes. Again, this is something we can test. This, we don't know. We're not actually looking at any genes, so we're guessing that that's how it will look. Now, to see how something's expressed in the population, if you remember your bio, right, your Mendelian genetics, so we would take a test cross, and we would take this homozygous parent and this homozygous parent. And we do that test cross in what we call the Punnett square. So the Punnett square looks like this, right? And we have this parent is here, and this parent is here, and we want to see how that trait is expressed. So when we do that, we have a big T and a little t. We have a big T, a big T and a little t. We have a big T and a little t, and a big T and a little t. So this shows that all of them are going to be medium tasters. But now when we want to see in the next generation, we need to now do a test cross with the heterozygous, right? All the offspring are this. So now these offspring are going to reproduce. They're going to have children. So I'm just going to erase this one. And now this is our next generation, right? Our heterozygous parents. A big T and a little t crosses with a big T and a little t. And this should be representative of how we see the trait in a population, and that includes in this class, in the class results. So I have my Punnett square again set up, and here we go. This parent, a big T and a little t. This, by this parent, which is a big T and a little t, and that gives us a dominant trait. That gives us a dominant but heterozygous trait. That's another dominant, and now we see the recessive trait. So when we look at this in a population, now we know that tasters to non-tasters are 1, 2, 3 to 1, right? And when we look at it in the population, we can break it down further, and we could say 25% of the population will be a super taster, 50% of the population will be a medium taster, and 25% of the population will be non-tasters. So this is, again, for sensitivity to taste. So when we look at this, we would think, well, if it's this low of a percentage, having the ability to taste, is that then going to be an advantage or a disadvantage if we see that in most of the population? So the test that you guys are going to be doing is to see where you sit here, right, and where the class sits. And we're going to be putting all those numbers together to try to figure out if we fall in with the expected results in the population.